Hai and Assalamualaikum. My name is Siti Zahira binti Ridwan.
Hai and Assalamualaikum warahmatullahi wabarakatuh. My name is Nurul Najah binti Murad. So today we are going to present about the mark-assisted selections in plant breeding. Let's start with the first part, which is me. Hello everyone, today I will be discussing mark-assisted selection, MAS, a powerful process used in plant breeding to select plants that carry desirable traits based on genetic markers. Unlike traditional breeding, which selects plants based on visible traits, MAS uses genetic information for more precise selection. In traditional breeding, we rely on observable traits which can be influenced by many factors and may take generations to develop. In contrast, mark-assisted selection allows us to look directly at plants' DNA, speeding up process and improving accuracy. There are different types of markers used in plant breeding, which is the first one is morphological, biochemical markers, and DNA markers. Morphological markers are based on phenotype analysis, which involves observable physical traits like color and shape. Biochemical markers involve gene product analysis, focusing on proteins and enzymes, and DNA are specific sequences in the DNA linked to the desirable traits, making them highly reliable for selection. Molecular markers are a type of DNA markers and play a crucial role in MAS. So these markers specific sequence of DNA associated with particular traits in plant, and there are segments of DNA with identifiable physical location on chromosome, and their inheritance can be tracked. Molecular markers arise from naturally occurring changes in polymorphism, the DNA sequences such as deletion, substitution, and repeat duplication. All molecular markers correspond to specific genomic position within the chromosome, known as log key, allowing breeders to pinpoint and select the best plants efficiently. In summary, mark-assisted selection leverages molecular markers to make plant breeding faster, more precise, and more effective, ensuring we can develop better crop varieties to meet the growing demand for food sustainability. So we move to the next presenter, Siti Zahira.
I want to tell you about importance of molecular markers. Let's dive into the fascinating world of molecular markers and their incredible impact on plant breeding. This tiny genetic tool brings amazing precision to the process, allowing breeders to pinpoint plants with exactly the right traits. Imagine the efficiency boost as we can now identify desired genes early on, making the breeding process faster and more efficient. Now, I would like to tell you about example of molecular markers. Now, let's explore some cool example of these markers. Simple sequence repeats, which is we call it as SSRS, are like nature's repeating pattern in DNA, and they help us spot genetic variation which is it. Then we have single nucleotide polymorphisms, which is we call it as S and P, tiny changes in DNA that are super powerful for detecting even the smallest genetic differences. How awesome is that? To wrap it all up, marker-assisted selection, which is MAS, brings so many advantages to the table. It boosts accuracy, slashes breeding time, and greatly increases our chance of developing successful new plant varieties. MAS is a game changer in creating crops that are disease resistant, high yield, and resilient to climate challenge. With molecular markers, we're paving the way for a brighter, more sustainable future in agriculture. That's all from our group. Thank you.